Mitochondria power the cell by converting ADP and phosphate into ATP. In this video, I'll give you an overview of mitochondrial structure and function. Here's the outer membrane of a mitochondrion, the inner membrane, which has many folds to increase surface area, and I'm just showing a few. The innermost region is the matrix, where most of the enzyme and most of the activity takes place. Between the inner and outer membrane is the intermembrane space. That's going to be very important when we talk about moving H+, creating a proton concentration gradient. The outer membrane has holes, pores or porins, and even medium-sized molecules can go through. These spaces are big enough for peptides up to about 50 amino acids. So much of the contents of the intermembrane space has about the same concentration as contents of the cytoplasm, but the porins are small enough to keep out most proteins. Contents of the matrix are much more tightly regulated. One of the most important aspects of matrix function is all of the enzymes involved in the citric acid cycle, which breaks down pyruvic acid or pyruvate from glycolysis into carbon dioxide and ultimately water. That requires NAD+, which gets converted to NADH in the citric acid cycle. That NADH binds to complex 1, a protein complex of electron transport chain. We'll talk more about that later, but I'm showing the major elements here. NADH binds and gives off 2 electrons and releases NAD+ and H+. And that feeds back into the citric acid cycle. The 2 electrons move through complex 1, Q, 3, C, and 4. The 2 electrons combine with 1/2 oxygen and 2 H+ to form water. That requires oxygen or the process stops. But what's really important about the electron transport chain is that the movement of those 2 electrons powers movement of a larger number of H+ across the membrane into the intermembrane space. And if you want to remember, it's about 10 H+ for each NADH. That creates a concentration gradient, which of course is pH, because this is H+. The pH becomes more basic in matrix because we're removing H+, and more acidic on the outside, pH 7, but it's actually not acidic. Of course, pH 7 is neutral, and it's about the same pH as the cytoplasm. That process of moving ions across the membrane makes the inside, the matrix, minus 150 millivolts compared to the intermembrane space and the cytoplasm around it. That means H+ has a strong tendency to enter. It's both more abundant outside and the inside is negatively charged relative to the intermembrane space. And so H+ will move through their only path back in normally, which is through ATP synthase. ATP synthase binds ADP in a phosphate. When H+ goes through, it takes about 3 to do this full cycle. It causes ADP and phosphate to be rammed together and form ATP. So ADP and phosphate enter, and ATP leaves the mitochondrion. The citric acid cycle is also producing carbon dioxide.